coming to the next one, steps in protein synthesis process or steps in translation process. See, already we know the steps in DNA replication and transcription process. See, the very first one, DNA replication is initiation, elongation, termination. In transcription also, initiation, elongation, termination. Both the cases, 3 steps are there, right? In the translation process also, initiation is there, elongation is there, termination is there. These 3 steps are also seen in the protein synthesis or translation process, right? See, before these 3 steps, okay, 2 more steps are there. Here, activation of amino acids. Next one is the charging of tRNA. Activation of amino acids, charging of tRNA. See, activation of amino acid means, see, any bond formation energy is required. See, amino acids are ready to react. Amino acids are ready to peptide bond, or one of the amino acid is there. The amino acid is activated. Then if amino acid is activated, then peptide bond formation takes place. What I am telling here, amino acid contains whatever that amino group is there or carboxyl group is there. These are activated. These are activated. Once these are activated, then these are involved in the peptide bond formation. Not only that one, see, the protein synthesis is a very, very specific process. Means one of the mRNA contains the codons. The codons are recognized by anticodons. Anticodons codes per specific amino acid. Remember, codons are codes per specific amino acid. Codons codes anticodons. Each anticodon having specific amino acid. That is nothing but codon. This is the one of the mRNA structure. There is in here. This is one of the codon UUU. This is called as codon. Codon codes for amino acid. We know how codon codes for amino acid. See, this is one of the tRNA structure. This is the tRNA. Okay, anticodon loop. Within anticodon loop, anticodon region is there. This is UUU. Within anticodon region, A is present. See, this A anticodon. We know UUU is codes for phenylalanine. UUU is codes for phenylalanine. Then the codon UUU codes for phenylalanine. The anticodon A is codes for phenylalanine. Codon phenylalanine. Anticodon here A. This one is codes for specific amino acid phenylalanine. Right? So that is the difference between codon and the anticodon. So codons codes for amino acids. Codons are recognized by anticodons. Anticodons which are present within tRNA, and specific anticodon loop or specific anticodon, okay, codes for specific amino acid. Specific anticodon codes for specific amino acid. That is nothing but here specific codon. Okay, specific codon codes for amino acid, and specific codon recognized by anticodon. Anticodon, okay, the specific anticodon codes for specific amino acid. Simple. What I am telling? Codons codes for amino acids. Specific codon codes for specific amino acid. How? Each specific codon recognized by anticodon. Anticodon codes for amino acid. Specific amino acid. Right? So that is about the codon and the anticodon interaction. So here, activation of amino acid means here all amino acids are activated, right? See, this process is very, very specific here. The total translation process is very, very specific process, because see, one form of message translated into the another form. Means whatever that RNA sequence, ribonucleotide sequence is there, the ribonucleotide sequence translated into amino acids. That is nothing but proteins. Sequence of ribonucleotides are translated into specific amino acid sequence. Specific amino acid sequence. That amino acid sequence are nothing but proteins. So here, activation of amino acid, charging of tRNA. The next step is charging of tRNA. Here charging of tRNA is nothing but this is one of the clover leaf structure of tRNA. Within this tRNA, we already discussed there is in different arms are there. This is anticodon loop, right? Here there is an amino acid acceptor arm is there. Means amino acid is binded at 3' side, at 3' hydroxyl group side, right? So if amino acid is present, if any tRNA contains amino acid, that tRNA is called as charged tRNA. That tRNA is called as charged tRNA. Means this is the tRNA, binded with amino acid. This is the tRNA binded with amino acid. So the process is called as charging of tRNA. See, specific amino acid bind at the amino acid acceptor arm to the tRNA. That process is called as charging of tRNA. Then initiation means 30S ribosome subunit binding and 50S ribosome subunit binding, right? So the process called as initiation. Here elongation is nothing but codon, whatever that codons are there, the codons recognition process. See, this is the one of the mRNA. This is the first ribosome complex, and this is the entire structure of the mRNA. Ribosomes are moving on mRNA. So if the ribosomes are moving on mRNA, the process is called as elongation. And next one is the last one, stop. The protein synthesis stopping process is called as termination. So there is an initiation, elongation and termination. These 3 steps, totally 5 steps are there. These 3 steps are common steps. These 3 steps are common steps for DNA replication and transcription and the translation. But here there is 2 more steps are extra for the translation process, right? First step, activation of amino acids. How the amino acids are activated, we know. Within the cell there is 20 types of amino acids are present. These 20 types of amino acids are involved in protein synthesis process. The 20 types of amino acids are present in protein synthesis process. This is the one of the cell and this is the nucleus, and whatever that for suppose this is one of the endoplasmic reticulum. Here ribosomes are present. There is an amino acid pool is there. Means all 20 types of amino acids are present here. This is called as amino acid pool. From this place to protein synthesizing site. This is the protein synthesizing site. So from amino acid pool to protein synthesizing site, amino acids are transported by the tRNA. So the very first step is activation of amino acids. So here 20 types of amino acids are there which are involved in protein synthesis process, which are activated. So in that, amino acids are react with ATP. So if the ATP hydrolysis takes place, the energy is taken by amino acid is activated, right? So when the ATP hydrolysis takes place, that time amino acid tRNA synthetase enzyme is present. That amino acid tRNA synthetase enzyme forms amino acid AMP complex. Amino acid AMP complex means amino acid and ATP. Between these two, interaction takes place. Presence of amino acid tRNA synthetase enzyme. So here amino acid AMP enzyme complex means how many number of components are present within this complex. One is amino acid. Amino acid is present here, right? And next one is AMP is present here. Next one is enzyme is present. 3 components are present. One is amino acid. One is the amino acid. And next one is the AMP. Right? This is the first one. This is the second one. And next one is the enzyme. Totally 3 components are present within that complex. Amino acid, right? AMP and enzyme. 3 components are present. and pyrophosphate is liberated. Pyrophosphate is liberated, right? So generally ATP, okay, we know generally ATP hydrolyzed or break down into ADP plus PA. This is adenosine diphosphate. Adenosine diphosphate. And this is the inorganic phosphate. This is the inorganic phosphate. But here ATP break down into AMP, adenosine monophosphate plus pyrophosphate. Plus pyrophosphate. This pyrophosphate is also very important. The pyrophosphate is also very important. I will explain why this one is important, right? So that is about that amino acid, AMP, enzyme complex. In this complex, three components are there. Amino acid is there, AMP is there, right? Enzyme is there, right? Here inorganic phosphate is there. Inorganic phosphate is there. This is one complex. This component is different from this complex, right? So that is about the first step, activation of amino acids. Activation of amino acids, right? Yeah. In the first step, we discussed about the activation of amino acids, and coming to the next one is the charging of tRNA. That is nothing but loading of tRNA. Loading of tRNA means this is the one of the empty tRNA. This is the one of the empty tRNA structure, right? See this tRNA structure, right? This is one of the DHU arm. This is T-C arm. This is anticodon arm or anticodon loop. Anticodon arm or anticodon loop, right? So this is the one of the tRNA structure. The tRNA, this tRNA is empty. When the amino acid, this is the 5' and this is the 3'. 3' hydroxyl group is available. 3' hydroxyl group is available, right? This is 3' hydroxyl group is available. Especially the 3' hydroxyl group. That hydroxyl group interact with amino acid. The 3' hydroxyl group interact with amino acid. So this is loaded tRNA. This is loaded tRNA or charging of tRNA. The process is charging of tRNA. Okay, this is called as charged tRNA or loaded tRNA. The process is called as charging of tRNA or loading of tRNA. Loading of tRNA, right? So this is about that tRNA, right? And parts are there. This is the anticodon arm. This is dihydrouracil. Okay, T-C arm. T-C arm, right? So here this is one of the DHU arm, anticodon arm. Remember, I like to ask one question here. Sir, how tRNA carries specific amino acid? How tRNA carries specific amino acid? We already discussed. The tRNA contains anticodon. Anticodon arm contains anticodon. Anticodon arm contains anticodon. That anticodon decides. The anticodon decides. Okay, which amino acid have to bind here? Which amino acid have to bind here? So a specific anticodon supporting to bind specific amino acid only. Specific amino acid only. For suppose here anticodon is, this is the one of the RNA. Here RNA sequence is UUU. This is UUU, right? And the anticodon, this is the anticodon. Here anticodon, this is the anticodon, right? Reason that anticodon is here A, right? For suppose here A is present. Here A is present within anticodon region, right? A is present, right? That A only supporting for the specific amino acid. The specific amino acid is here specific amino acid is here phenylalanine. The specific amino acid here phenylalanine. So here what I am telling, specific codon recognized by anticodon, right? Each specific anticodon codes for specific amino acid. Each specific anticodon codes for specific amino acid. That is nothing but specific codon codes for specific amino acid by specific anticodon. So between these two there is an interaction is there. If anyone asks me the question, sir, how mRNA codes for specific amino acids, I will say the answer, mRNA contains codons. Codons are recognized by anticodons. Each anticodon interact with specific amino acid. Then mRNA contains sequence or specific sequence or specific codons codes for specific amino acids. That's the reason why, see, the protein, different types of proteins are there. Different types of proteins means different types of amino acid sequence is there. Different types of amino acid sequence are there. So the concept is very simple. All of you remember, the mRNA contains codons. Codons are recognized by anticodons. Each anticodon codes for specific amino acid. If anticodon is changed, specific amino acid is changed. If anticodon is changed, specific amino acid, changed. See, when the anticodon is changed, if codons are changed, anticodons are changed, anticodons are changed, amino acids are changed. Amino acids are changed. So here codons are codes for, simply we are saying codons are codes for amino acids, specific amino acid. Codons are codes for specific amino acids by the anticodon. And one of the important concept is amino acid tRNA synthetase enzyme. Amino acid tRNA synthetase enzyme means, sir, here specific anticodon is there. For suppose AAA, this is the specific anticodon. How specific anticodon interact with specific amino acid only? How the tRNA have the capacity? To interact with specific amino acids. For suppose here we have the 61 codons, means sense codons, total 64. In that 61 codons are there. 61 codons are codes for 20 amino acids. 20 amino acids, right? So based on this one, 61 codons are there. 61 codons are there and 61 anticodons are there. 61 codons are there. 61 anticodons are there, right? So here 61 codons are there, 61 anticodons are there, and these 61 anticodons are codes for 20 amino acids. These are codes for 20 amino acids. So how the specific interaction is takes place? How specific anticodon, codes are attached with specific amino acid, by the amino acid tRNA synthetase enzyme. We'll discuss how the interaction is takes place, right? How the attachment is takes place, right? So here, how the specific amino acid attachment takes place. Here the attachment is completely depends on amino acid tRNA synthetase enzyme, right? See, this is the one of the amino acid tRNA synthetase enzyme, right? It's having two active sites. The enzyme having two active sites. Generally enzymes are having only one active site, right? But this enzyme is quite different. And the enzyme is having two active sites. See, this is the one of the active site 1 and this is one of the active site 2, right? In active site 1, specific amino acid is attached. Generally we know in a cell 20 types of amino acids are present. 20 types of amino acids are present. In the same manner, in a cell 20 types of amino acid tRNA synthetase enzymes are present. In a cell 20 types of amino acids and 20 types of amino acid tRNA synthetase enzymes are present. Each amino acid tRNA synthetase enzyme each amino acid tRNA synthase enzyme is very specific with the amino acid. For suppose this is the amino acid tRNA synthase enzyme. This enzyme only interact with the glycine. So how the specific interaction is takes place? For suppose we know about this is one of the structure of the glycine, right? This is the H and this is also H. Generally this is the side chain, right? This is the H. This is the amino acid glycine. This is the amino acid glycine, right? So the glycine only fit within the enzyme active site. Within the enzyme active site, because this structure only fit within the enzyme active site. Within the enzyme active site. So how this enzyme differs remaining other amino acids? For suppose if we this is the structure of CH3, right? This is the H and this is the COOH, right? And this is the structure of alanine. This structure of alanine. Glycine, right? The first structure is the glycine. Here H is present. This is the second structure. This is the amino acid alanine. This is the amino acid alanine. So structurally differentiation is there. For suppose this is the amino acid tRNA synthase enzyme. The enzyme interact with only glycine. So this structure, this amino structure only fit within the active site of the enzyme. Okay, this structure is not fitted because here H is present, here CH3 is present. So based on the structural differentiation, the active site structures also differ. That's the reason why we already discussed within the enzymes. Specific enzyme interact with specific substrate. Glucokinase enzyme only interact with the glucose because glucose has a particular structure. Here also same thing. See, one of the amino acid tRNA synthase enzyme, this enzyme only interact with the glycine. So interaction between glycine and amino acid tRNA synthase enzyme is very specific. For suppose if alanine comes to here, the alanine amino acid not fit in the active site, because this enzyme only interact with the glycine. For suppose alanine interacting amino acid tRNA synthase enzyme is there. So within that case, that active site fit for the alanine amino acid. So within the cell, 20 types of amino acids and 20 types of amino acid tRNA synthase enzymes are present. So the tRNA synthase enzyme and the amino acid interaction is very specific. The amino acid interaction is very specific. Right? So that is about the first active site. Here having two active sites are there. One of the active site. So within the cell, 20 types of amino acids and 20 types of amino acid tRNA synthase enzymes are there. Amino acid tRNA synthase enzymes are there. So the interaction is very specific. Specific amino acid specifically interact with amino acid tRNA synthase enzymes. Right? Then that is about that first explanation. That is the explanation regarding to the active site 1. This is the explanation regarding to the active site 1. And coming to the next one, this is the second active site. We already discussed the enzyme having two active sites. This is the best example, tRNA synthase enzyme. The tRNA synthesis, this is one of the tRNA structure. How the loading takes place. See, we already discussed the tRNA having 3 arms. This is the anti-codon arm. This is the anti-codon arm, and this is T size C arm, right? Exactly here, 3' hydroxyl group is present. Amino acid is attached. Amino acid is attached, right? So remember all of you, once the specific amino acid fit within enzyme active site, then the DHU arm, this is the DHU arm. So there is an interaction is present, DHU arm and the amino acid tRNA synthetase enzyme, and the amino acid tRNA synthetase enzyme. See, so this is one of the amino acid tRNA synthetase enzyme, right? This is the enzyme, active site 1 and active site 2. So how the specific amino acid added to the tRNA? We know always there is a specific interaction is there between the codon and the anticodon. Codon and the anticodon. All of you remember the specific amino acid addition. Specific amino acid addition here. First of all, this is the active site 1, right? And this is the active site 2. All of you see the amino acid 1. Active site 1. This is the amino acid. Particular amino acid is fitted here. And coming to the next one, the active site 2. Active site 2. Within this case, see the DHU arm. The DHU arm, okay, of tRNA is interacted. The DHU arm of tRNA is interacted. Remember, between amino acid and anticodon, between amino acid and anticodon, between these two, matching is takes place. Both these are matched. Anticodon and amino acids are matched. The DHU arm also matched. The DHU arm also matched. See, between these two, the amino acid and anticodon, both these are not matched. The DHU arm also not matched. The DHU arm also not matched. So, the specific amino acid addition to tRNA depends on, okay, first of all, amino acid active site. See, the amino acid tRNA synthetase enzyme, that is the active site 1, and the next one is DHU arm. Next one is the DHU arm, because see, if the amino acid, again listen, this is a very little difficult topic and this is a very important topic, right? See, amino acid 1 and anticodon, see, both these are matched. DHU arm also matched. Both these are not matched. DHU arm also not matched. Then if the matching is takes place, the amino acid, okay, if the matching is takes place between these two, matching is takes place, this one also matched. If the matching is takes place, the amino acid is added. The amino acid is added. Right. The amino acid is added to the tRNA, especially at the 3'. This is the 5' position, right? This is the 3' hydroxyl group position. Always in molecular biology, the 3' position is very important. The 3' position is very important because here hydroxyl group is available. So the 3' hydroxyl group provides site to bind the amino acid. Once the matching is takes place, whatever the amino acid is there, once the matching is takes place, this one is also matched, this one is also matched, then the binding of amino acid to 3' position. That is also called amino acid acceptor arm. All of you, again, this is the total explanation. Charging of tRNA. Okay, that is also called loading of tRNA. The total this process is called as charging of tRNA or loading of tRNA. This is charged tRNA. Means, okay, the tRNA bind with the amino acid. The process is called as charging of tRNA or loading of tRNA. This is charged tRNA. Right? So during this process, specific amino acids activation, because the cell contains that 20 types of amino acids are activated, and these are specifically interact with specific amino acid tRNA synthetase enzymes. Then amino acids are attached to the tRNA. See, the interaction especially depends on two steps, because here the enzyme contains the two active sites. One of the active site, okay, the first active site, okay, specifically interact with the specific amino acids, and the next one, the next active site of amino acid tRNA synthase enzyme interact with the DHU arm of tRNA. DHU arm of tRNA. So the simple concept is, if the amino acid anticodon is matched, the DHU arm also matched, then binding of amino acid to amino acid acceptor arm, right? So that is about that charging of tRNA or loading of tRNA, right? Yeah, this is the second step overview. In that case, in the first step we already discussed. This is the complex. This is the complex formed within the first step during activation of amino acids, amino acid AMP, right, enzyme, amino acid AMP enzyme complex, right? See, that is the first complex formed within the first step. In the second step, this is the tRNA structure. See, the tRNA not attached with any amino acid. The tRNA not attached with any amino acid. So, after the completion of second step, amino acid tRNA enzyme plus AMP separated, right? Plus the tRNA attached with the amino acid. The tRNA attached with the amino acid, right? Before the second step and after the second step. Before the second step and after the second step. See, after the second step, see whatever that amino acid is there, the specific amino acid attached with the amino acid acceptor arm. Amino acid acceptor arm of tRNA. Amino acid acceptor arm of the tRNA, right? So that is about that overview of the second step. Within that case, within this case, the very first complex, is the first complex formed within the first step, right? How the interaction takes place here. This is the tRNA, right? So after that interaction, amino acid tRNA enzyme and AMP, and next one is the tRNA with amino acid. This is also called charged tRNA. Charged tRNA, right? So that is about the second step. That is about the second step. That is nothing but loading of tRNA or charging of tRNA. In the next class, we will discuss about the initiation, elongation and the termination. Remember this topic, especially the protein synthesis is very difficult topic. In NCERT also not given up to this level, but this concept is very, very important, right? All of you, please watch this video second time or third time also, because the protein synthesis or translation process is very difficult. So in the next class we will discuss about the initiation, elongation and the termination.